Well, thank you very much for hanging in here until the very end. I'm very intimidated to have to give a talk after such an impressive talk from the Kingsley Group, obviously, just fascinating. You're paving the way, and it's very intimidating. Thanks a lot to the organizer as well, Aki and Wolf, for the invitation. I really enjoyed the meeting, very enjoyable. And I had so much food and wine that I probably won't have to eat for a whole week when I get back home. So I'm sure it's pretty much the same for a lot of people. Okay, so this talk was put together with a former PhD student of mine, Sébastien Renaud, is now a postdoc in Lauren Risberg lab. So we left the fish for sunflowers. All right, so it's already 10 years ago that Dolph in his famous book wrote that knowledge on ecological causes of adaptive divergence and speciation had made substantial leaps over the last decade and allowed identification of the most pressing research areas. So we saw some, many talks that progress have been moving on as well since that time on ecological processes involved in adaptive radiation. But until last year, he could still write that one of the most glaring deficiencies is the almost complete absence of information on the genetics of ecological speciation. So I think we just saw in this talk that things are moving quickly, so that's where we are at this point. So some of the several unresolved key questions that still have to address are: what is the contribution of ancestral genetic variation and new mutations to the evolution of restricted gene flow? What is the relative role of regulatory and structural variation? What is the relative role of various molecular mechanisms, sense and transregulation, allele-specific expression, alternative splicing, epigenetic control in the differential pattern of phenotypic expressions? We are very in the infancy of addressing those things. And finally, something that has been going on in the literature, has been stirred a lot of interest in the last couple of years, is what are the size, distribution, and nature of genomic islands of divergence that are being maintained in the face of gene flow in the process of adaptive divergence and ultimately leading to reproductive isolation. So that mainly referred to the metaphor of genomic islands of divergence that was first proposed by Taylor and Al in 2005, and that refers to the phenomenon that locus that are being under the effect of strong directional selection by tight linkage will maintain chromosomal areas of strong divergence relative to sea level, which correspond to the upper limit of expected neutral divergence. And such regions will alternate with regions of purely chromosomal regions evolving under purely neutral process. So, if you want the seafloor. So that has some analogy of also with the concept of or the genic view of speciation that was proposed by Chong Wu back in 2001, whereby adaptive divergence involved heterogeneity of genomic divergence during population divergence and speciation, and whereby differentiation accumulates in some regions, whereas gene flow precludes divergence elsewhere. And as the process goes on in time, then these regions can grow into larger genomic islands of divergence via divergence hitchhiking. And ultimately, there is a complete reproductive isolation, and you get to the stage where you can talk about, I guess, real and real species without any hope of return. So recently also Sarah Villa proposed a verbal theory of speciation via divergent hitchhiking, whereby to propose a mechanism whereby this progress of increased size of genomic island of divergence could occur, and she proposed that this was associated with the reduction of interpopulation recombination due to selection against immigrant alleles that would ease speciation in the face of gene flow and allow the increase of size numbers of islands of divergence. And this will depend on the size of genomic regions that are being affected. So to date, there's really mixed empirical evidence for that. So first, there are some empirical evidence for large regions of differentiation being maintained in the face of gene flow, coming from the work on her own work on the PFs, and Sarah suggested that regions all the way up in the orders of several centimorgans could be maintained in this process, and some work from Pat Nozzle also in walking sticks and others. But there are also evidence for small regions of differentiation, not as having nothing to do with large regions of differentiation, and barely reflecting the concept of genomic islands of divergence. So clearly, it may depend on the possibility that this may happen, this process may happen or not happen, may depend on the specific context. And that was recently investigated by Pat Nozzle and Mark Feather, whereby through analytical approach, they investigated the efficacy of divergent hitchhiking in generating genomic islands of divergence during ecological speciation. So they were mainly interested in investigating the relative effect of effective population size, selection coefficient, migration rate, as well as recombination rate on the extent of differentiation that can be achieved and the size of genomic islands that can be maintained. So their main conclusions, let's get some water. So their main conclusion is that divergent hitchhiking may sometimes create a large region of differentiation around selected sites, but this is under very specific conditions that they could observe that. So mainly you really need reduced recombination locally, but higher recombination rate otherwise. This can happen in the context of low migration rate among populations, so they define low migration rate in the order of 0.001. Small effective population size, so in the order of 1,000 and smaller, and a relatively strong selection coefficient. And also the probability of effect, they show that the probability of effect will increase with the number of loci under selection, which is consistent with the multifarious selection hypothesis of a famous paper by Rice and Oster in 1993, whereby speciation is promoted by a multitude of different selection pressures acting on many genes and phenotypes. All right. So some predictions that you can make from this concept is that the development of genomics island of divergence will be eased by a phase of allopathic divergence. That increase in size and number of genomic islands, size and number of genomic islands should increase with the extent of adaptive phenotypic divergence being reached between populations. And with time, genomic islands may become submerged via a high genome-wide level of differentiation. So as the process progresses, on average, there will be a higher level of differentiation being reached between populations such that such island of divergence may not become obvious anymore. And this phenomenon needs to be investigated in early stages of population divergence. So that brings me to our whitefish system. So just to give you some background information on the evolutionary history of these populations. These populations occur in northeastern North America, and the history of it is that there has been a phase of allopatry of geographic isolation in distinct glacial lineages that our best guess is that relates to two glacial events ago. So the Illinoisan glaciation event, whereby there was a phase of geographic isolation in allopatry, and but without any specific phenotypic or average phenotypic divergence being obvious of fish belonging to one or the other glacial lineages. Then following the last glacier retreat, there has been secondary contact occurring between the two lineages, so following the last glacial retreat about 12,000 years ago. And so there has been secondary contact in a series of lakes, and in some of those lakes, but actually a small proportion of those lakes, through the process of ecological opportunities and competitive interactions, one of the lineage got a derived phenotype to adopt what we call the dwarf phenotype. And these things are partially reproductively isolated, and then gene flow being maintained between them. And so they differ in very important ways in many phenotypic aspects. So in terms of morphology, they have morphological specialization associated with either a benthic or planktonic mode of life. In the broad sense, there are very distinct life history characteristics that we can broadly define as the benthic being a K specialist as opposed to a R specialist for the limnetic. Strong differentiation in terms of bioenergetic efficiency, whereby to reach a same size, the limnetic will have to feed much more intensively, and therefore, in terms of growth rate in control conditions, you get a higher growth rate in benthic as opposed to the planktonic. And then in terms of swimming behavior and activity, these fish are slow mover, and with other distinct characteristics, whereas the limnetic are they have a high swimming activity type of life. So all of that can be summarized as a case of life history trade-off between growth and fecundity associated with the normal forms versus survival function associated with the limnetic form for occupying distinct niches. Okay, so then so that's 6 numbers that summarize 4 years of work. Basically, it's just a brief summary of transcriptomic work whereby we studied genome-wide gene expression differences between the forms at different life stages. So essentially, that's one of my favorite results. It shows that even if you do genome-wide transcriptomics, null results can happen. So basically, there's no difference at the embryonic stage in terms of overall gene expression between limnetics and benthics, but in a matter of weeks, when you get to the juvenile stage, about 10% of the genes are differentially expressed, and same thing later on at the adult stage. And overall, if you make a summary of that in terms of associate the gene expression with functional, different biological functions. Essentially, genes that are overexpressed and they overrepresent the following biological function in the dwarf phenotypes: energetic metabolism, muscle contraction, detoxification, lipid metabolism. That, to make a short story, that we associate with survival functions, whereas biological functions that are overrepresented by genes that are overexpressed in the normal form are associated with cellular cycle and growth and protein synthesis, so growth functions. And then based on the different gene expression differences between the forms, you can easily tell apart a dwarf from a normal fish from its overall physiology, if you want. Okay, so then to start to make the link back to the pre-requirement for to assess genomic islands of divergence. So this here shows that in terms of some of the demographic characteristics, these populations can comply the requirements for hoping that genomic islands of divergence can develop. So roughly speaking, our best estimates of effective population size on average for any of the dwarf normal population that we analyze in these systems are in the order of 1,000. And migration rate between dwarf and normal in each specific lake is generally small, but highly variable, with some lake having a higher migration rate or gene flow between them, and all the way to a very, very reduced migration rate between population. And a migration rate negatively correlate with the extent of phenotypic divergence between the forms in each of these 6 different lakes, which led us to propose that the intensity of selective pressure being acting specifically in each of the lake was driving the extent of phenotypic divergence and limiting or reducing gene flow to achieve that in the different lakes. So essentially, we proposed a long time ago that this continuum of divergence could be associated with a continuum of selection intensity. So we have done, I'm not too much of an ecologist, but we have done a little bit of what I call evolutionary limnology. So we went into pretty much in the detailed analysis of the physicochemistry of those lakes, as well as investigating documenting the planktonic and epibenthic prey community. And essentially what we got out of that is that lakes with higher level of phenotypic divergence are characterized by less habitat availability, whereby lakes are shallower and they go into hypoxia in the deeper water in summertime. So there's reduced habitat available. Less resources, smaller prey range size and biomass, and on average a wider gap between planktonic and limnitic prey size. So overall, the proposal that we make is that landscape with higher potential for intra-population competition and more potential for distinct trophic niches is associated with increased divergence and reproductive isolation. So then we have been doing quite a bit of gene mapping work in order to to investigate the genetic basis of some of the traits that are of particular interest in the adaptive divergence of those populations, and also to get some identity put some identity to anonymous markers that then we were interested in screening in the wild population to document the extent of divergence. So overall, we built a map with low-resolution map with about 800 markers, but we could identify several QTL associated with phenotypes that are of particular interest in the divergence between these forms, things related to growth, swimming behavior, condition factor, and gill rakers. But also we use expression, gene expression as phenotypes, and we search for expression QTLs for genes. So we ended up finding over 500 expression QTLs that involve 375 different genes. And then you can have an overlap between both, and that's what we call quantitative trait transcripts. So basically, an expression QTL can co-localize with a phenotypic QTL at the same chromosomal regions, and that's what you define as quantitative trait transcripts, as Greg Gibson defined it. For example, I'll give you just a quick example of that. So an expression QTL for the troponin genes that is involved in fast muscle contraction overlap in localization at the resolution that we had with the burst swimming phenotypic QTL. Okay. So then those same markers that were used to build a map were screened in 4 different benthic limnitic pairs to look for markers under selection by the general genome scan framework. But then the first difficulty was to get the homology between the markers used to build a map and then finding homology of the markers in the natural population. So that's a low-resolution genome scan. But still, at the end, we ended up identifying overall in the 4 lakes that we investigated 24 outliers. That was based on AFLP analysis. But since we had some information from the map, we could get some identity to those markers. So out of the 24 markers, we can put an identity on 19 of them. So some of them were either only associated with one of the phenotypic QTL, or only associated with expression QTL. And so these numbers here refer to the fact that you can have, very often we had co-localization of expression QTL for different genes at the same chromosomal region. So we end up considering only one, all of that as a single eQTL. And then you have QTT, whereby you have an overlap of pQTL and eQTL. Okay, so more recently then we tediously developed a handful of SNPs annotated with known functions in order to further investigate the role of natural selection in driving divergence in those populations. And so we screened these and we developed those SNPs from cDNA sequencing by 454. They were validated, and then they were screened in 5 different sympatric species pairs. And so basically, in the general sense, they kind of confirm what we already knew from AFLP and microsat data, is that the level of genetic differentiation between limnitics and benthics vary from one lake to the other, going with very small, very little differentiation up to very high differentiation in Cliff Lake. And this totally followed the gradient of phenotypic divergence that we documented in those lakes. So this is quite congruent. Then we perform lake-by-lake genome scan analysis in order to identify outliers in the SNPs in the 5 different lakes. So to make a brief summary of that. So what we observed is the same thing. Well, there was a gradient in the proportion of markers that we could label as outliers from the lake of low differentiation to the lake of highest differentiation. So the proportion of markers that were potential outliers, so markers that were potentially under the effect of directional selection, went from 0% to 15% depending on the lakes. So, and that again goes in the direction of the extent of phenotypic divergence that we observe between those forms, which suggests or support the fact that the intensity of directional selection acting in each of the lakes vary along a gradient. So an example of one of these outliers, triose phosphate isomerase, which is a key gene in producing an anaerobic energy through the glycolysis. So this gene is a strong outliers in Cliff Lake, and through some association mapping, not in the QTL mapping, but in just looking for association of variation with fish from back-cross families that have all the variants of phenotypes covering the limnitic benthic phenotypes. So basically there's a genotypic association between variation at this gene and phenotype that is more like a limnitic type as opposed to benthic type of fish. And this gene is also differentially expressed systematically in all of the lakes that we have investigated between the dwarf and normal. So that's, for us, that's quite a cool candidate genes to do further investigations here. So that's one of my favorite results out of these recent investigations. So what it shows here is the level of observed heterozygosity, and that's only for Cliff Lake, so the lake of highest differentiation, in normal and dwarf. So observed heterozygosity, in blue you have all the markers, the non-outlier markers, and in red, the Cliff Lake outliers. So what you see is that there's a really sharp reduction of heterozygosity only for outliers in the dwarf population. So actually, out of the 12 outliers that we identify in this lake, 10 got to fixation in the dwarf population, but there was no significant reduction for these same marker in the normal population. So the heterozygosity of these markers just about the same as the background markers, if you want. And same thing, the background markers in the dwarf population is not, the heterozygosity is not significantly different than the other ones. And then we compare the level of heterozygosity of the variation of these markers with populations that are representative of the ancestral variation of this Acadian lineage from which the dwarf cliff originated. So essentially, these markers here have nothing special in terms of reduction in variation in the ancestral genetic pool. So it really suggests that strong directional selection have been responsible for driving the fixation of these markers, but only in the dwarf phenotype. So there's a strong asymmetric direction of selection operating in the system as suggested by those results. So that's a different way of representing that. That's the same result I just presented for Cliff. We observed the same significant pattern in Webster, reduction of diversity with some fixations only in the dwarf, in the outlier markers for the dwarf, nothing special with the normal, and then non-significant trend of reduction of heterozygosity in Indian Lake and East Lake. And these markers are not, so this is not a case of very strong evidence for parallel evolution. Outliers being involved in one lake are not necessarily the same of the other lakes. So we're talking about reduction of diversity and fixation on different genes from one lake to the other. Okay, so it seems that we have some of the ingredients for genomic islands of divergence to be operating there. So this is a recent divergence event in terms of the phenotypic divergence in the system following the phase of secondary contact. This divergence over a very vast areas of co-adapted and integrated physiological traits and functions. These populations are characterized by generally small effective population size and small migration rate, although variable. And I think we have evidence, although we don't have any quantification of selection coefficient, evidence for strong but variable directional selection. Okay, so where does that lead us? So this is all, what I'm going to show you now is very preliminary. It was done like one day before I got on the plane, so don't tear off my head when I present that. So what we did here to investigate if there could be an effect of selection acting on QTL outliers, on the differentiation of non-outliers markers through the effect of divergent hitchhiking. So we just documented through a graph the relationship between the extent of the level of genetic differentiation of different markers for which that we could investigate in natural population as a function of the chromosomal distance from closest QTL outlier QTLs. And we did that for the different type of QTLs that we have. So I'll show you an example detailed example from Indian Lake. And that's for the phenotypic QTL outliers. So basically what we have is a significant negative relationship between the level of FST values and the chromosomal distance from the closest outlier QTLs. Same thing for the expression QTL, same sort of pattern, highly significant negative relationship from in the same direction. Much less resolution for QTT outliers, but still the trend is a negative relationship, but this was not significant. When you put all the QTL types together, so you end up with this highly significant relationship between reduction of FST values with the chromosomal distance from the outliers. So the bottom line here is that you would not expect, by just a random process, you would not expect this reduction or this linear negative relationship from the outlier as you go away from the outlier if there was not some hitchhiking effect associated with selection acting on the outlier. So that's the main point. So then, if we look lake by lake, considering all QTLs together, and more or less going from the 4 lakes we investigated, going from the lake with the least phenotypic differentiation to the lake with the most pronounced level of different phenotypic differentiation. So this is the the head off part of the talk. So again, for East Lake, same thing, we got the strong negative relationship and significant relationship between FST and distance from the nearest QTL outliers. So it's very hard to try to define the actual size of genomic island of divergence out of that. But let's just put it as a relative basis to compare from one lake to the other. If we consider the level of the upper FST value of neutral markers, and we just make the correspondence, that defines a region in the order of many centimorgans. But this is just to do some relative comparison. In East Lake, we have this highly significant relationship overall as well, but the slope is much milder, so we end up with the mean FST being encountered in a much further away. For the second lake with the highest divergence, Webster, same thing again, significant negative relationship with mean FST values coordinating at 25 centimorgans. And then in Cliff Lake, again, highly significant negative relationship with the mean FST value getting to the slope at to the curves at 50 centimorgans. And linear relationship was the best fit for this distribution of that. We tried negative, quadratic, and so on, and we didn't get a better fit than linear. Okay, so we find then if we go through all the information I presented and make some characterization of the genomic islands of divergence and comparing the different lakes, again going from a lake with least differentiation, phenotypic differentiation, to the lake with the highest level of phenotypic differentiation. So what we saw with the SNP outliers is that there's an increase in the proportion of SNP outliers from East Lake to Cliff Lake. So totally independent of the SNP information. Let's say we define the number of islands based on the QTL outliers distribution that we just documented. There's some sort of a trend in an increase from in the number of islands, at least from least differentiated to the most differentiated. So at the very least, the least differentiated pairs have the least number of islands and high proportion of islands for Cliff Lake. In terms of size, that's very relative and very arbitrary. So if we just consider as the standard the distance that was coordinating the mean FST values that I just presented, and we use 1 as the standard for East. So basically, again, we go in terms of increase in size, a trend of increase in size from East Lake to Webster and Cliff, Indian being a bit of the odd one. Then if we define the relative sea level for each of these lakes, the sea level would be if you want, the FST outlier threshold that would define outlier markers from neutral expectation. And basically again going like the sea level kind of increase from the least differentiated lake to the most differentiated lake. So the threshold to define outliers, FST threshold to define outliers in Cliff was all the way up to 0.46, which is very, very high. So you would expect from what I presented at the beginning that this could be a case where the actual islands would get submerged and you would not detect them. But it's totally the opposite actually. So if you define the height of the island by the difference between the mean FST value of outliers in a lake minus its sea level, you get the height of the island in this particular lake. So again, you have a trend of increase of height of the island from East Lake to Cliff Lake. So which saying specifically, for example, that the mean level of FST values above the threshold is much higher in Cliff Lake than it is in East Lake, even if the threshold is much higher there too. So the outlier here are more outliers in the lake of most pronounced differentiation than they are in the lake of least pronounced differentiation. To me, was kind of unexpected. Okay. So in summary, adaptive divergence between dwarf and normal whitefish definitely involves many co-adapted and integrated physiological traits and function. So I think the way to find many phenotypic differences between whatever is to look for them. That's a good start. So I think we have been investigated a lot of phenotypic traits and expression traits and so on. So basically, you end up with realizing that there's a lot of things that make the difference between those animals. Divergence seems to be maintained by strong selection acting on many QTLs and genes that are associated with the expression of adaptive traits that are distributed throughout the genome. Those populations are characterized by small effective population size and migration rate, and numerous divergence QTLs make divergence hitchhiking potentially effective over. This is to be, have to be cautious with that, but right now it definitely suggests that large chromosomal regions can be under the effect of this hitchhiking divergence process. But of course, this phenomenon needs to be investigated with much more increased resolution, which I hope next time I make this, present the whitefish story, will integrate some of those new results. We're just gathering raw data right now, like we get into the rad tag frenzy, like many of you, in order to hopefully increase by 100-fold the number of markers to revisit our map again with a much higher resolution and redo our genome scan again, and to refine and retest all of those things with a higher resolution to see if we're totally off the track, or if there's some reality in just what I just told you. So but that will generate only anonymous markers, or for the vast majority. So as Caval presented yesterday, we are very interested also into the exon capture sequencing techniques, and we are currently developing a SNP based on the exonic coverage of about 3,000 annotated genes specifically for whitefish, in order to screen for variation specifically in coding region. Okay, since I'm 1 minute, because I have to tell you before I leave. I always have to do that, so I have to leave you with reminding you or letting you know, if you're not aware of it, of the existence of this journal, Evolutionary Application, that was launched in 2008, and a journal that's the first journal and the only journal that really specialized in the use in paper that make use of evolutionary concepts and or methods to investigate issues of applied issues of relevance to many human affairs, either in agriculture, related to climate change, invasion biology, evolutionary medicine, and so on. I have to tell you about that journal because just happened I'm the editor-in-chief of it. And we are fortunate to have a great team of associate editors and editorial advisory board. Andrew and Isabel are part of the team. And the journal is endorsed by SSC and ESEB as well. We are the only evolutionary biology journal that applied double-blind review process. There's optional open access, and we just got our first impact factor last year, and we started not too bad, ranking us up as a 9 in evolutionary biology journals. So please submit your best relevant papers to us, and if you're very fine looking, you may end up making the cover pages. Thank you. Say that again?
You mentioned that the two species are partially reproductively isolated.
Yeah.
What are the barriers, and do they vary among lakes? For instance, do they follow along with divergence?
There's well, definitely. I mean, all of those lakes where these things thrive. We're talking about very small lakes, so there's no geographic or physical barriers and there's no spatial barrier to them. They totally overlap in distribution. So reproductive barriers has to be, we have not investigated but it's most likely has something to do with pre-zygotic isolation. That could be related, that's the classic in fish is size assortative mating, which could make a lot of sense in those things. Other than that, there's nothing to prevent them to totally mix in the lake.
So, I have a question that is in comparison to Felicity Jones, the one from top to yours. You were finding these genomic islands on the order of 5 to 57 morphs. Felicity was finding them on the order of 10 to 50 kb. I'm wondering if you could comment briefly on that comparison.
Yeah, I think I can comment. I'm like you. I'm not too sure if I should believe these results, but it just happened that, I mean, systematically we see that negative relationship. So that's a bad start. I mean, if you go back to Pat analytical investigation of that, I mean, you see that if you change parameters values, you can quickly go to the possibility of occurrence of this to a totally flat expectation that nothing will happen. So it could well be that, well, there's different things. That first, I'm not too sure about what's the extent of divergence overall in the anadromous freshwater system, if these things are speciating or not. And the other thing is that, I mean, I think we can fairly guess that you can have huge effective population size and much higher migration rate in the sticklebacks as opposed, and maybe it's just demographically, you don't have the setup for this to happen. But still.
One more question. Yes, I would like to ask if you would consider this an example of coevolution.
Oh.
If you would consider this an example of coevolution, I mean, if both species...
Of coevolution?
Yes, if both species influence each other, or if it's one species which is more influenced by the other without ever
Yeah, I don't know. We never thought about that sort of system as a system of I mean, somewhere, somehow, I guess they co-evolve, but how's one form influence the fate of the other? I'm not too sure how to comment on your question.
When you were talking about the continuum of divergence and increasing the percent of outliers, could there an effect of the high average FST values in the test for looking for outliers? Or
The effect of the high FST values?
the average FST is high
Yeah.
because of the high FST of a lot of flows and
Yeah.
very high FST of quite a few.
You mean in terms of generating this apparent continuum? Is that what you're saying?
No, what I'm saying is like in these four populations that
Yeah.
you can see an increase in the percent of outliers.
Yeah.
You can see an increase in the average FST.
Yeah.
And the number of outliers you can detect can be different just because the FST is not
Yeah,
higher.
but no, that's a good point. But I intuitively, before seeing those results, I would have thought that the total opposite effect, in the sense that if your threshold is lower, you should expect that it's going to be even easier to detect outliers. If you get your threshold upper, intuitively, I thought it would be less likely to be able to detect markers that would be above that threshold. So for one thing, so I think if it would have an effect, would be the other way around. And on top of that, I mean, the difference between the mean FST values of outliers, it's even higher. From the outlier threshold to the mean values of outliers, it's even higher in the lake of high differentiation. So I think the possible artifactual effect would be going totally in the opposite direction.
Any more questions? Not
Time to go.
Luis and Yara,